Basic greetings. Today we will be reading the book of Carl Zimmer, The Planet of Viruses, 2nd edition, 2015. Chapter The Alien in the Water Cooler. Giant Viruses and the Definition of Life. Whatever there is a water on Earth, there is a life. The water may be in a Yellowstone geyser, in a pool, in a cave of crystals, in a cooling tower sitting on the roof of a hospital. In 1992, a microbiologist named Timothy Rowbotham scooped up some water from a hospital cooling tower in the English city of Bradford. He put it under the microscope and saw a welter of life. He saw amoebiae and other single-celled protozoans about the size of human cells. He saw bacteria about 100 times smaller. Rowbotham was searching for the cause of outbreak of pneumonia that had been raging through Bradford. In the ranks of the microbes he found in the cooling tower water, he thought he found a promising candidate, a sphere of bacterial size sitting inside amoeba. Rowbotham believed he had found a new bacterium, the doubted Bradford coccus. Rowbotham spent years trying to make sense of Bradford coccus to see whether it was a culprit in the pneumonia outbreak. He tried to identify its genus by finding matches with genus in other bacteria of species of bacteria, but he couldn't find any. Rowbotham had reached a dead end by 1998, when budget cuts forced him to close his lab down. He wanted to destroy his buzzing Bradford coccus. He arranged for French colleagues to store his samples. For years, Bradford coccus languished in obscurity until Bernard Lascola in Mediterranean University decided to take another look at it. As soon as he put Rowbotham's samples under the microscope, he realized something was not right. Bradford coccus didn't have the smooth surface of spherical bacteria. Instead, he was more like a saucer ball, made up of many interlocking plates, and Lascola saw hair-like threads of protein radiating out from its geometrical shell. The only things in nature known to have these kinds of shells and threads were viruses. But scientist Lascola knew, like all microbiologists in the time knew, that something in the bacteria of Bradford coccus was 100 times too big to be a virus. And yet a virus is exactly that Bradford coccus turned out to be. When scientist Lascola and his colleagues examined it further, they discovered that it reproduced by invading amoeba and forcing them to build new copies of it. Only viruses reproduce this way. Scientist Lascola and his colleagues gave Bradford coccus a new name to reflect its viral nature. They call it mimivirus in honor to the virus' ability to mimic bacteria. The French scientists set out to analyze the genes of a mimivirus. Rowbotham had tried and failed to match its genes to those of bacteria. The French scientists had better luck. The mimi virus turned out to be virus genes, and a lot of them. Before the discovery of mimivirus, scientists had become accustomed to find only a few genes in virus, but mimivirus had 1018 genes. It was if something took the genomes of the flu the cold, smallpox, and the 100 other viruses, and stuffed them all inside one protein shell. The mimivirus even had more genes than some species of bacteria. In both its size and its genus, mimivirus had broken cardinal rules of being a virus. Scientist Laskola and his colleagues published the details of their remarkable mimivirus in 2003. They wondered whether it was unique or whether there were only giant viruses also hidden in the plain sight. They collected water from cooling towers in France and added amoeba to it to see whether anything in the water might infect them. Soon the amoeba were exploding, releasing giant viruses. They were not mimiviruses, however. They were another species with 1,059 genes, setting a new record for the biggest genome in a virus. While the new virus looked a lot like mimiviruses, its genome was profoundly different. When the scientists lined out the new virus's genes with those of mimivirus, they could not match for 833 of them, but the another 226 were unique. The scientists decided the new giant virus needed a name. They called it mamavirus. Other researchers joined in the hunt of the giant viruses, and they began finding them everywhere, in rivers, in oceans, in lakes, buried under Antarctic ice. In 2014, French researchers thought out pieces of Siberian permafrost that had been frozen over 30,000 years ago. They discovered giant viruses in the found soil at 1.5 micrometers long. They were the biggest giant viruses yet found. Scientists have ever begun finding giant viruses lurking in animals. Scientist Laskola and his colleagues collaborated with Brazilian scientists to study serum samples taken from mammals. They found antibodies in giant viruses in monkeys and cows. The researchers have also isolated giant viruses themselves from people, including a patient with pneumonia. It's not clear yet what role giant viruses play in our health, though they might be able to directly infect our own cells, or they might just lurk harmlessly in amoeba that invade other bodies, or even our bodies. The story of giant viruses drives home just how little of virus fear had been explored. It also injects fresh life into long-standing debate. What began over a century ago with the discovery of viruses? What does it mean to be alive? Before scientists discovered viruses, they generally agreed on the definition of life. Living things could use their metabolism to survive, grow, and reproduce. In other states of matter, like clouds or crystals, might seem alive in one respect or another, but on the whole they didn't come close to meeting all the standards of life. When scientist Wendell Stanley produced crystals of tobacco mosaic virus in 1935, he unsettled those neat distinctions between the living and the non-living. As a crystal, his virus behaved like ice or diamonds, but when provided into tobacco plant, it multiplied like any living thing. Later, as the molecular biology of viruses came into a crisper focus, many scientists decided that we were only lifelike, but not truly alive. All the viruses that scientists studied carried a few genes apiece, leaving the wide genetical gulf dividing them from bacteria. The few genes that viruses carried allowed them to execute the various tasks required for making new viruses, to invade a cell and slip their genomes into cells' biochemical factories. Missing from viruses were all the genes for full-blown life. Scientists could find no instructions in a virus for making a ribosome, for example, the molecular factor that turns RNA into proteins. Nor did viruses have genes for enzymes that break down food in order to grow. In other words, viruses appeared to lack much of the genetic information required to be truly alive. In a theory, though, a virus might be able to gain that information and become truly alive. After all, viruses are not fixed in stone. A mutation might incidentally duplicate some of their genes, creating new copies that could take new functions. A virus might accidentally take up genes from another virus, or even from the host cell. Its genomes could expand until it could feed, grow, and divide on its own. While it's conceivable that viruses could evolve their way to life, scientists saw a giant wall in the way. Organisms with big genomes need a way to copy them accurately. The odds of suffering a generous mutation increase in a genome gets larger. We protect our giant genomes from this risk by producing error-correcting enzymes, as do other animals, plants, fungi, protozoans, and bacteria. Viruses, on the other hand, have no repair enzymes. As a result, they make copying errors in a tremendously high rate, but we do, in some cases, over 1,000 times higher. The high mutation rate of viruses may put a limit on their genomes, and thus they prevent them from being truly alive. If a virus's genome gets bigger, it's more likely to suffer a lethal mutation. Natural selection may therefore favor tiny genomes in viruses. If that's true, when viruses became unable to make room for genes that would let them turn raw ingredients into new genes and proteins, they cannot grow, they cannot expel waste, they cannot defend against hot and cold, they cannot reproduce by splitting in two. All these knots added up to one great devastating knot. Viruses were not alive. An organism is constituted of cells. The microbiologist scientist Andrei Lvov declared in a lecture he gave when he accepted a Nobel Prize in 1967. Not being cells, viruses were considered little more than cast-off genetical material that happens to have the right chemistry to get replicated inside cells. In 2000, the International Committee of Taxonomy of Viruses made the judgment official. Viruses are not living organisms, they flatly declared. But in the years following this declaration, many virologists came to question it, and some rejected it outright. In the face of new discoveries, the old rules no longer work out and work well. Giant viruses, for example, went overlooked for so long in part because they were 100 times bigger than viruses were supposed to be. They are also loaded with far too many genes to fit old-fashioned notions of a virus. Scientists didn't know that giant viruses do all of their genes, but some suspect that they do some rather lifelike things for them. Some genes in giant viruses encode enzymes that can repair DNA. They may use these enzymes to fix any damage. They occur while traveling from one host cell to another. And then, when giant viruses invade amoeba, they don't dissolve into a cloud of molecules. Instead, they set up a massive, intricate structure called a viral factory. The viral factory takes in raw ingredients through one portal and then split it out new DNA and proteins through to others. Giant viruses may use their viral genes to carry out at least some of this biochemical work. The giant virus's viral factory, in other words, looks and acts remarkably like a cell. It's more like a cell, in fact, that the scientist Laskola and his colleagues discovered in 2008, and it can be infected by the viruses of its own. This new kind of virus, which they named a bacteriophage, slips into the virus factory and fools it into building virophages instead of giant viruses. Drawing dividing lines through nature can be scientifically useful, but when it becomes to understanding life itself, those lines can end up being artificial barriers. Rather than trying to figure out how viruses are not like other living things, it may be more useful to think about how viruses and other organisms form a continuum. We humans are an unexpected blend of mammal and virus. Yes, remove our virus-driven genes and we would die in the womb. It's also likely that we depend on our viral DNA to defend against infections. Some of the oxygen we breathe is produced through the mingling of viruses and bacteria in the oceans. That mixture isn't a fixed combination, but an ever-changing flux. The oceans are a living matrix of genes, shuttling among hosts and viruses. While it's clear that giant viruses bridge the gap between ordinary viruses and cellular life, it's not yet clear how they reached that ambiguous position. Some researchers argue that they started out as ordinary viruses and stole the extra genes from their hosts. Other scientists have counted The giant viruses started out early in the history of life as free-living cells and became more virus-like over billions of years. Drawing a bright line between life and non-life doesn't just make it harder to understand viruses. It also makes it harder to appreciate how life began. Scientists are still trying to work out the origin of life, but one thing is clear. It didn't start suddenly in the flicks of a great cosmic power switch. It's likely that life emerged gradually as raw ingredients like sugar, phosphate combine in increasingly complex reactions on the early Earth. It's possible, for example, that single-strand molecule of RNA gradually grew and acquired the ability to make copies of themselves. Trying to find a moment in time when such RNA life abruptly became alive just distracts us from the gradual transition to life as we know it. The RNA world, life, may have consisted of little more than fleeting coalitions of genes, which sometimes thrived and sometimes were undetermined by genes that acted like parasites. Some of those primordial parasites may have evolved into the first viruses, which may have continued replicating down to the present day. Scientist Patrick Forte, the French biologist, has proposed that in the RNA world, viruses invented the double-stranded DNA molecule as a way to protect their genes from attack. Eventually, the host took over their DNA, which then took over the world. Life, as we know, in other worlds, may have needed viruses to get its start. As long last, we may return into the original two-sided sense of the word virus, which originally signified either a life-giving substance or a deadly venom. Viruses are indeed inquisitively deadly, but we have provided the world with some of its most important innovations: creation and destruction joined together once more. Thank you for your attention. It was the chapter of Carl Zimmer book A Planet of Viruses, 2nd edition, 2015. Thank you for watching, and press like, write your comments and ask your questions, and subscribe to my channel on YouTube. Bye bye.